Yes.
Okay, so in this area is where the lumen is, and these are the layers of the stratified epithelium of the esophagus, and these will be in the submucosa, these areas. So when you go deeper, you're going to find the muscular layers deeper in this histological structure. Now, when you go lower, you'll find that now you are no longer in the esophagus. At this point, at this juncture, you are within the stomach. And usually it is the first portion of the stomach wall. So you can see the layers of the epithelium have become single layer, because all you have to do is look at the number of nuclei and you see it's a single layer of nuclei. And then the cells are columnar as opposed to what you have here. So you usually have first the cuboidal, then they become flattened, and finally the ones at the end, they are keratinized squamous, and that's why they end up being fluid. But here we have columnar cells, simple columnar. And then secondly, we are going to start seeing that what you don't find within the esophagus are indentations of the epithelium forming secretory tissue. We don't have that. But in the stomach, we are going to see all these indentation of the epithelial lining into the mucosa. And these indentation, they form tiny tube-like structures, which we call the gastric glands. And they start producing stuff which they release into the lumen. However, in the esophagus, we do see, I'll go back, I think I'll skip a few slides here. Here, I wanted you to see this structure. In the esophagus, if you can see this diagram, what we have where my arrow is pointing is the lumen. And with this lumen, I have now my stratified layer here. Now what happens is that deep in the submucosal layer, we do have some tissue, a lot of lymphoid tissue, and this helps in the immune system. Number two, we are going to see that we have some few areas where we have a lot of mucosal glands. These ones that are indented here, they are not as distinct as those of the stomach. Those ones, they produce the mucus. So they're in those areas. So I wanted you to see the organization of, and I think I'll start with this, then I'll go back to the diagram of the mesenteries. In the last diagram, I told you that we have 4 layers that are common to 4 layers that are common to all parts of the GIT, starting from the lumen, because in this tube-like structure, it's part of a GIT. This area is the lumen, so it has been cut from here to here is a longitudinal section, and this is a cross section that is not completely severed, so it is cut halfway, or 1/3 of it, and that 1/3 is removed so that we can see the lumen inside. Then towards the end, the whole part has been cut. So this is a cross section. So we have this part being a cross section and this region being a longitudinal section. Now, what I wanted you to see here are the 4 layers that we are talking about. The innermost next to the lumen is what we call this mucosal layer. Then there is a thin lining of the muscularis, the mucosal muscularis here, or I would say just a thin layer of smooth muscles. This layer separates the mucosa from the submucosa. So next to it, we are going to see now this pale blue structure here being the submucosa. You can see here. And the submucosa has some blood vessels and it has some glands. Now, from the submucosa, the next layer we are going to see is the circular muscles. And you can see that in this area where I have a longitudinal section, the circular muscles have been cut and you can only see their cross section. However, in this circular or cross section, you can see the elongated cells which are arranged in a circular manner. Next to it, we have the longitudinal muscles. So these two make up the muscularis externa. Now, surrounding that, we have now the serosa here. And the serosa is made up of the epithelial cells, which secrete some serosal fluids so that they help in lubrication. Now, what I wanted you to take note of is the arrangement towards the lumen. You can see the mucosal layer, how it has high infolds. These are called the circular folds or placa circularis. Like I told you, this is in a foreign language, which I don't care so much for. All you need to remember, these are circular folds. And what you need to take note of, these circular folds are permanent. So they are not going to stretch out. They are permanent circular folds. So how do we form this? I would like you to imagine if you have your foot here and you put on a sock. Say, if I put your sock here and then I decide to pull it down so that it ends up having those folds and going down like that. And on this other side, the same. Imagine that on this, you're going to wear a boot. So you take your boot and you wear it here. So what you'll have is a smooth boot on the outside because it is straight. But when you look at your sock, it has all these infolds. And if I were to follow one of these folds, it forms a circular form. And this is what you have here. This is like the sock that is towards the inside. Your leg here represents the lumen. So you have all these infolds, but surrounding them we have the straight tissue here making up the muscles and the serosa, and we'll discuss the mesenteries later. So with that imagination, you imagine that those folds of the socks are permanent, and between the straight portion and the folds here, this is where we have the mucosa. So the socks is representing the epithelial layer plus the mucosal layer, but then we have the submucosa between them, and then we have the smooth muscles. Now, if you have that in mind, the next thing I want you to remember is that if you take this serosa, it's a very thin membrane, single cell layer of epithelial cells, which are also secretory. They appear, these layers appear like a cling wrap. If you have that transparent cling wrap paper that you use in the kitchen, if you take one of the items, say for example, you take your rolling pin, okay, you have your rolling pin there, and you take a cling wrap, and in this cling wrap, you take one sheet as it appears here, then you roll it around your rolling pin so that it comes out from this other end, and you flatten it out here. So now you have an area with a double layer here, and then you have a single layer of membrane surrounding the pin, and this is what we have here. So you have your serosal membrane here. It comes, it rolls around your GIT to the other end, and then it comes and flattens against the same sheet of membrane. Now the interesting thing is that as it forms, it traps the blood vessels between these sheets. So you have your blood vessels are trapped here. At the same time, we also have your nerve tissues, if I represent them with green, trapped between those two layers. So you find that the nerve tissues will follow the blood vessels and they'll keep branching, and then they'll end up coming to branch into the intestinal walls. It is important because these two layers of the sheets form the mesenteries. They are the ones which hold your intestines so that they don't fall off. Otherwise, if we didn't have the mesenteries holding the intestines in place, every time you wake up, you start walking, all your intestines will settle to the bottom of the peritoneal cavity. But this does not happen because these mesenteries are attached to the body wall, and as they are attached to the body wall, they prevent your intestines from falling, and that's what we have here. So in the organization of the GIT, I needed you to look at the 4 layers of the GIT, and then secondly, look at the organization of the mesenteries. So if I were to cut a cross section of your abdominal wall, you can see this diagram here. If I cut a cross section with your vertebral column towards the dorsal side, what you're going to have at the front is the peritoneal cavity, and you find that your intestines are hanging inside this peritoneal cavity. On the one side of the peritoneal cavity, we have the parietal peritoneum, and then the one that is surrounding your intestines is the visceral peritoneum. And this is representing now the serosa, where the two come together and they form a double membrane, they form a mesentery. And the mesenteries are the ones that are holding those intestines in place. So if you look at the next diagram here, this is just the gross structure. I think this one you're familiar with. When you go to the lab, this is what you see. But if you look at the schematic representation, we have your intestines here. But towards the inner portion, we have now the mesenteries here. If I were to cut a section through your body here, this is the schematic representation, and you can actually see the mesenteries here. This will be the mesenteries, and this is cross-section of your GIT surrounded by the serosa. So I wanted you to have that idea in mind so that when you have the blood vessels, they are going to come and distribute themselves among these double layers of the mesenteries, and then they will terminate in the abdominal or the GIT wall. Any questions so far? So again, this is just a representation of part of the GIT, showing you the arrangement of the mesenteries, which look like the transparent cling wrap that I have just described. And these fatty areas, the areas with a lot of fat, you can see that right in between we have blood vessels. And running parallel with the blood vessels are these white fibers which represent the nerves. So they run together and they innervate, and they actually they supply blood to the GIT. So from there, I think I can now move on. This diagram, I think I had discussed it last time, but I will use it as a reference every time I go to a different part of the GIT. So last time we discussed the esophagus and we saw how thin it is. How thick it is, not thin. It is important to know the thickness and to look at the layers of muscles that are involved here. Within the esophagus, Remember, this is where we swallow, where we need peristaltic movements to push down a food bolus. And the food bolus has just been formed from the mouth, made up of solid food. So it requires high powerful muscles to contract and push down the food bolus. So if you look at the muscles of the GIT, remember we are discussing the longitudinal muscles and the circular muscles. You can see that the thickest layer is between the esophagus because it's the one that requires to push that food towards the stomach. This is also another schematic diagram. I have quite a number. So this will show you now the 4 layers the way I have described them, showing you a single layer of epithelial cells. And with these epithelial cells, you can see that they are columnar epithelial cells. Towards the lumen, they have some cytoplasmic extensions, which we call the microvilli. Now, within some of these epithelial cells, you can see that they have this indentation open area. These cells are different from the columnar cells. These are usually the ones which will secrete mucus and it will come into the lumen. Now, from the epithelium to the muscularis mucosa, this is the single layer of circular muscles. We have now, this will be the mucosa, epithelium plus this connective tissue. The connective tissue between the epithelium and the muscularis mucosa represents the lamina propria. So all that will make up the mucosa. The next layer being the submucosa, which is rich in large blood vessels and nerve fibers, plus a lot of contractile proteins and a lot of fibrous proteins, making a denser connecting tissue as compared to the lamina propria. And as you see, you move further out, you are going to go to the circular muscles and then to the longitudinal muscles. And then finally, we go to the serosa. At this point, I also want to highlight that we have 2 other layers. Remember, I told you I'll start increasing the complexity of the 4 layers. First, we describe the 4 layers. Then we start increasing complexity by bringing a web structure or a web arrangement of neural tissue. So this arrangement is usually found between the submucosa and the circular muscles, and we form what we call the Meissner's, or you can say the submucosa plexus. This plexus is a network of neurons which form part of the enteric nervous system. Then we have another web or layer of neural tissue which lies between the circular muscles and the longitudinal muscles. And this is known as the myenteric plexus. My represents the myo for muscles. Enteric represents the GIT. So it's the plexus of neurons that is seated between the circular and the longitudinal muscles, or between the muscle layers. So myenteric plexus between the muscle layer. Submucosal plexus sits between the submucosa and the circular muscle. The two communicate and they help in motility. So when it comes to the submucosa and the mucosal plexus, they usually communicate with one another and they help in initiating contraction of these muscles. So we'll come to that later. Let's see. Now that you have seen the organization, these are just diagrams showing you the mesenteries again. So This is just a review slide, but I can ask a question. How each of the areas of the GIT are modified to perform their function. So you can have modifications in the mouth, modifications in the esophagus, or modifications in the stomach. So I will skip these and go to the next. I'll leave you to look at the questions. So from this slide, you can see we have covered what is in the mouth and dentation. We've covered the pharynx and the esophagus, and within this we have looked at the swallowing reflex and the involvement of the swallowing center of the brain. So I'm hoping that all of you are following what I'm saying. Are you in class?
Yes.
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 Okay. And am I clearly audible? I'm using internet from campus, so I don't know whether you can hear me clearly.
Yeah, it's clear. We can hear you properly.
Yes,
Okay.
it's clear.
Okay, great. Murunga ka Kenya,
Yes.
you can hear me. I'll
Yeah,
start
I can hear
names
you.
randomly so that people don't lose.
Yeah.
Lucy Kenya. Lucy, are you there?
Yes, I'm here.
Okay, great. So I'll just call people's names randomly as I talk so that you pay attention. I don't want to lose you as I talk. So the next region I'm going to discuss is the stomach. So when I leave the esophagus, I go to the stomach. Separating the two is a sphincter known as I think I had mentioned it in class last time. So Madeline, can you tell us that sphincter or that valve that separates the esophagus from the stomach? What was it called? Madeline, can you remember?
Yes.
What was it called? It has two names actually. One
It's
representing the function and the other one location. So who can remember? Madeline, you
esophageal
said you can remember one name. Tell us.
the esophageal sphincter.
Lower esophageal sphincter is one name. And I think the first person to put up their hand is Nicholas. What is the other name?
Cardiac sphincter.
Cardiac sphincter. So you can either call it the cardiac sphincter or the lower esophageal sphincter. Now, when you mention lower, you remember there is always the upper esophageal sphincter. So between the upper and the lower is the esophagus. So let us look at the stomach. Now, the stomach is a pocket. It's a holding house or room. Whatever you eat, if it were to go straight into the GIT and into the intestines to be digested, you feel so full or you've been pushing food before they are ready to be moved. So immediately you swallow your food, it has to be held in a holding room or a holding pocket. And this pocket happens to be the stomach. The shape of the stomach as it is a J shape. It has that curved area which can enlarge. It can increase in size and it can shrink. Number two, you find by looking at the stomach and looking at the types of cells that are there and even the function, you can divide anatomically the stomach into several regions. And the main ones, you can see that they are color-coded in this diagram, where the pink is representing the esophagus and that area will be the cardiac sphincter. Immediately after the cardiac sphincter is a region known as cardia. Just from the name cardiac, so you get cardia. It is the region next to the cardiac sphincter. Now, like I said, it's a J shape. So the top part of the stomach acts like an umbrella, and it is a fundus. This area is the one that collects the gas. When you are digesting food, you may be producing some gases, and the gases will collect to the top. Now imagine if this stomach was completely coming from the esophagus and enlarging into a stomach like that. It means every time you produce the gas will accumulate at the cardiac region, and you'll always be feeling like eliminating that gas, making you produce gas or belch all the time. However, there is this modification of the stomach so that the bulge is forming a place where it can capture the gases, trap them, and when you are ready to release most of them, then you can belch and all these gas comes out. So the function of the fundus is to collect the accumulated gases that are formed or those that are swallowed. Even when you swallow your food and you find that the air comes in, it will bubble up and come to the fundus. From there, we have the body of the stomach. And this is where most of the parietal cells and those, the cells that produce hydrochloric acid and those that will produce enzyme are formed. You find most of them will be formed around the body so that they keep producing the gastric juices. And these gastric juices will mix with the food so that you can start the digestion. As the food travels down, it will come to a cone-shaped region like that, so that the shape of this area is the pylorus. So this will be the pylorus. And it has the large part of the pylorus, which is known as the antrum. And then we have the cylindrical part of the pylorus at the end, which is the terminal pylorus or distal part. So the pylorus is divided into the antrum region and into this cylindrical pylorus. So in this area, what you're going to find again are mainly the cells that will produce the enzymes, and number 2, cells that produce hormones. So if you are to follow at the beginning here, the fundus, these are not mainly secretory. Those cells, they just form part of the wall, they're structural. As you move down, you go to the body, and what you start stimulating is production of hydrochloric acid. So we find a lot of cells that are related, or they are concerned with secretion of hydrochloric acid. But we also start seeing enzyme being produced. So we also have cells that are producing pepsin. Or if you remember, I said from last time, it is produced in form of an inactive protein called what? Who can remember?
Pepsinogen.
Pepsinogen.
Pepsinogen. So it is the inactive form. You have to cleave it to form the active pepsin. And you need the hydrochloric acid to cleave it and to activate it. Then as you move down, now you start producing hormones. And with the major hormone that we have is gastrin. This is the hormone. So the cells that produce gastrins are towards the pyloric region. In addition, we have a lot of mucus being produced. Now, generally, mucus are produced all along the walls of the GIT, starting from the mouth. Go to the esophagus, you always have cells that are producing mucus. But you find within the pylorus, we have high concentration of cells that are producing a lot of mucus. Now, what are the main functions of the stomach? The first thing I said, it's a pocket that will hold the food. And the purpose of holding that food there is so that it is released when it is ready to be released. When is it ready to move to the next step? When it has become liquefied. Remember, the food is coming in as solid. You have to crush that food, add gastric juices, mix it, tear it down until it forms a liquefied mixture. That liquefied mixture is a chyme, and when it forms the chyme is ready to be released into the next part, which is the duodenum. For that reason, the stomach is going to hold the contents until they are ready to move to the next area. Now, what holds them in place is this sphincter, the second sphincter, which is the pyloric sphincter. So you remember on the upper part of the stomach is the cardiac sphincter, the lower portion is pyloric, coming from the name pylorus. So, as the food is mixed, when the food is pushed towards the bottom of the stomach, towards the pylorus, this pyloric sphincter will open very slightly, and it will take only a second to open and close. Within that second, anything that is liquidish will be jetted out. So it will be spurted into the duodenum. And once the sphincter closes, you find that all the remaining food will be pushed back into the stomach. And then what will happen is the stomach will contract again and try to squeeze all the contents to come towards the bottom. And when they reach there, the pyloric sphincter will open slightly, again allow small amount of the chyme to go into the duodenum. For that reason, the stomach is also controlling the rate at which the food enters the duodenum. It is the one that holds the food there, and it will allow it to be emptied into the duodenum. And that process that I have just described is what we call gastric emptying. Now, the other thing that we have mentioned is that within the stomach we have a lot of acid secretion. We need that acid for two purposes, actually three. Number one, you need it so that it can cleave the pepsinogen and activate it into the active form of pepsin. Number two, that pepsin requires a very low pH in which it can act. And number three, you find that the acidic environment is hostile to any microbes that may have been swallowed together with the food. So this is one area of defense. Remember, we say the first area of defense is within the mouth, within the saliva, where we have the zymogens and we have the lysozymes, we have the immunoglobulins. All those are acting on they are helping in the immune function. Now, anything that may have escaped and goes into the stomach, as the food bolus is broken down, it will be mixed with the acid, and the acid is usually corrosive to the walls of the bacteria, and therefore it will destroy the bacteria. So it has some antibacterial action. The other thing I mentioned is that we have a lot of secretory activity, so we are producing a lot of gastric juice. This juice is added onto the food mixture, therefore making the food fluid. So, Asna, your hand is up. Can I hear what you have to ask?
Excuse me, madam, I didn't get the second function clearly.
Which function?
HCl, the second function, I didn't get it clearly.
The first function we said is to cleave the pepsinogen so that you release the active form of pepsin. The second function will be to provide an optimum pH for the pepsin, because the pepsin requires a very low pH. So it provides that environment for the pepsin to work. And you'll see that in the lab, when we raise the pH, when you use an alkaline media, the pepsin rate of reaction is greatly reduced or inhibited. So you need the low pH for the pepsin to be active. Then number three, it is the antibacterial action. whereby it destroys the walls of the bacteria and therefore rendering or destroying the bacteria, the microbes. Yeah?
Thank you.
Welcome. So the next function of the stomach is to cause the food to change from the solid particles into a liquid that can be moved into the duodenum. So remember we said that what is ejected out here is a fluid. And for it to have converted from the hamburger to a fluid, you require a lot of secretory activity within the stomach. So let us follow the food that you chewed in the mouth. So it was a solid piece of bread, and as you chew, you are cutting it physically and mixing it with saliva. Now, by the time it goes to the back of the stomach, it forms a capsule-like structure like this, which is able to go to slide through the esophagus. Now, this structure is not solid because remember you have chewed it and then you compacted it like this. So I tell people to use their imagination here. I don't know how many of you have made sand castles. Yeah, you go to the beach, or if you have sand at home, you take sand and you put water and you compact it and you form a ball. And then you decide you're going to form a house from it. Now it will appear solid, but all you require to do is to heat that structure and it disintegrates. So this is similar to the food bolas that you eat. Because it is made up of very, very tiny particles but are clamped together and compacted with saliva in between. Once it falls into the stomach, it will find a pool of fluid. And once the muscles of the stomach start to push it and hit it against the wall and to move it around, this structure disintegrates. And when it disintegrates, it leaves those tiny particles that had been chewed. And now you can have enzymes starting to work on them. Now, if they are not proteins, there will be no enzymes acting on it. However, the mechanical action of the stomach contracting and pushing these contents against a very tiny space. And then when there is no space to eject them, because we usually have the contraction starting about halfway around the stomach here. So we start some contractile activity, and this contraction goes like a ring. It's like you put a ring here and you are pushing it down. So as you push it down, it is forcing the contents to go towards the terminal portion. And because all these contractions are happening along the walls here, you find that most of the contents are going from the wall towards the bottom. However, when the sphincter here is closed, these contents are forced to come back through the central portion. So it is like from the sides, it hits the bottom here and it is released to form like, I would say, we see like a volcanic eruption so that it comes back here. By the time it's coming back, the stomach will have relaxed. So the fluid is pushed back. And then we have another wave of contraction that will push it to the end and then push it to the middle back to the front. So we'll have all this circular motion, and remember that the walls are contracting and pushing the food against it, so it will tear the food into smaller pieces. So those pieces that had not been chewed, because of that mechanical action of the stomach and the fluid that has been secreted, it will, within time, it will become all fluid, and therefore we say the stomach is making the contents fluid. So the second function is fluidization of the stomach contents. Yes, Asna.
Is it that movement? Is it what is called self-churning?
Just hold on a bit. I can hardly hear you. I don't know whether this will make it better. Okay, ask your question now.
I'm asking the movement of the food and the fluid. Is it what is called self-churning?
The chyme is the fluid itself.
And the churning, the one.
Oh, the churning, yes, that the mixing, the movement, all that is churning. You are churning it, you're mixing it up there.
Oh, okay. Thank you.
Okay. So, yeah, I have to reduce that volume because it's filling my ears. And when I don't, let me see. Oh, I can't reduce the volume as I talk. Yes, Asna, or you didn't lower your hand?
Sorry.
Okay. So the next function of the stomach is the preliminary digestion of the proteins. Remember we said that we have the first portion of proteins are digested within the stomach, and this is done by pepsin. But as we look at other functions, we are going to see that we also have some other enzymes like the gastric lipases. We also have things like renin, which is being produced. All those are enzymes that are produced here. So although we have lipases, you find that it's not a lot of digestion of lipids that take place there, but most of it will be protein digestion. So let us look at the anatomical structure. We have we have divided the stomach into several regions. So we have the cardia here, we have the fundus, we have that whole body here, and then we have the pylorus. Remember the pylorus I told you is like a cone. So we have the cylindrical part here, and then we have the wider portion. The wider portion is what is called the antrum, and the other one is more of the cylinder. In this case, they are calling it the canal. Now, what I need you to watch here is that if I cut the stomach and I remove the top layer, which is the serosa, I cut and peel off the serosa, I'm going to see the longitudinal muscles. Okay? If I pull off the longitudinal muscles, remember the four layers that we discussed. The one of the layers is made up of muscles, the outermost being the longitudinal muscles, and the inner one being the circular. So if I peel off the serosa, I'm going to see the longitudinal muscles. If I peel off the longitudinal muscle in this area here, you're going to see the circular muscles. Now, one of the unique things about the stomach as compared to other parts of the body is when you peel off the circular layers, you find a third layer of muscles, and these are called the oblique layer. So the stomach is the only organ that has 3 layers of muscles. Now, if you recall, we said that when you have circular muscles, if this is a tube and you have longitudinal muscles here, the work of longitudinal muscles is to shorten the tube. The work of circular muscles is to constrict or to make the lumen smaller. So using the longitudinal and the circular, you can enable the food to move in the longitudinal direction by peristaltic movements. However, in the stomach, we have these oblique muscles so that they cut diagonally. So let me use a different color, blue. So these ones will cut diagonally. So as the longitudinal muscles are contracting, they will shorten the size of the stomach. As the circular muscles are contracting, they will push the contents of the stomach to the bottom. But then we have longitudinal, which will move the food upwards and backwards. so that instead of going simply by peristalsis, you have the oblique, which can also cause waves within the stomach. So you find the stomach has modification that will allow more mixing rather than just simple peristaltic movements. So that is one modification. Number two, we have the innermost layer is highly folded as compared to the outermost layer. Now, if you remember the diagram I had just showed you, where you have things like the socks that are folded. Now, in the stomach, those folds are not permanent. They are called temporary folds here, as compared to the permanent folds that are found in the small intestines. These temporary folds are called the rugae. R-U-G-A-E, the rugae. So, what is the function of these temporary folds? As you fill in the stomach with the food contents, you find that it will find a very small pocket, and it will require this pocket to enlarge to be able to hold that big plate of food. I see that when you serve yourself dinner, you may have a flat plate, but you make a mountain of food. This mountain of food has to go somewhere. As you chew, it will enter into the stomach. It has to be held. And then when you finish that plate, you want to fill it up with a liter of fluid. Say, for example, your soft drink. And as though that is not enough, you decide you're thirsty, you also want to add water. Where are all these going to? They all end up in the stomach. So the stomach has to be elastic enough to accommodate all the food that is coming in. Now, for that to happen, the first thing, so that it does not create a lot of pressure, remember, if you have a small tube and you add contents there, the contents will exert pressure against the wall. So they'll be pushing against the wall. Now, if that were to happen, it means every time you take something very simple, like maybe just one biscuit, it will create a pressure that will make you feel uncomfortable and you'll stop eating. And yet that amount of food is not sufficient to give you enough nutrients. So what the stomach does, it has these temporary folds so that when you put in your food contents, these folds will flatten out. And as they flatten out, they increase the volume inside so that they will be accommodating more and more contents. Now, as though that is not enough, the walls of the stomach will also enlarge. They are elastic and they'll push out. So first, you flatten out the stomach lining. So these rugae will become, remember when you are hungry, they are highly folded. But as you increase the contents, they start becoming flat. And when you are very full, it is completely lined up like this. Then after you empty the contents, the rugae will start forming bulges because the muscles on the walls are contracting and pushing against that lining. And as you continue emptying, it will become highly folded inside here. So we have talked about the epithelia. I don't have to repeat. I've talked about mucus, acid, and pepsin. So the one thing I have not discussed is what cells secrete what. And this is captured in this box down here. We have talked about mucus being produced. What are the cells that produced? These are the surface cells within the epithelial lining. They have the ability to produce mucus, and this mucus is usually neutral. It doesn't have the bicarbonates, it doesn't have, because we need an environment that is acidic. So you cannot produce bicarbonates within those mucus. So it's mainly neutral mucus. Number 2, we have the parietal cells. Parietal cells are the ones which produce hydrochloric acid. The other name, because hydrochloric acid is highly corrosive, they are also called oxyntic cells. It's like they are causing oxidation and therefore they are causing corrosion. So remember, oxyntic and parietal cells are one and the same, and they are the ones that produce hydrochloric acid. The next one, remember we said pepsinogen. Pepsin is produced in form of pepsinogen. So zymogen is enzyme. Chief cells will produce pepsinogen. So remember parietal for HCl. Chief cells are also zymogenic cells. Zymogenic cells produce pepsinogen. Then we say that another function, especially in the pyloric region, we have a lot of gastrin or hormone production. So we have cells that are specialized to produce the hormone, and these are called endocrine cells. Remember endocrine because they produce hormones of the enteric system. So they are enteroendocrine cells. The enteroendocrine cells are the cells of the intestines that produce hormones. In the stomach, the major hormone being gastrin. So what does gastrin do? It stimulates the stomach walls to start producing HCl and to start producing pepsinogen in preparation for digestion. So when you start feeling you have food contents in the stomach, or you start stimulating gastrin production so that it can produce those two environments that are required for digestion within the stomach. I think this diagram is just to remind you the relationship between the wall, epithelial wall in the esophagus, and look at that one of the stomach. So the epithelial lining will come and form indentation deep into the mucosa and then come back to the surface. Again, deep into the mucosa and raise up to the surface. Now, if you look at the surface view, this area that I'm pointing, it appears like a smooth surface. So it appears like a sheet of smooth surface, but this sheet has tiny holes. And each of these holes is representing a gastric gland. So if I were to cut through half of the hole, I will find there will be an indentation into the mucosa, representing the gland here. So this is a three-dimensional structure where the surface is just showing holes or pores. But when you go to the edge where there is a pore, you find it has an indentation of the epithelial lining forming the gastric gland, and this is what you are seeing here. So these are the pores and these are the glands. Now you can see even within the esophagus, you had these pores that are extending from the submucosal gland, and this is one of them. Remember, it is going to the submucosal area, and they are the ones which produce mucus within the esophagus. In the stomach, we have the gland. So I'll go now to the stomach here. This is just a block representation of the stomach. So what we have here, I told you on the surface you appear like you have those pores. But if you cut through one of those pores, you form that they indent into a gland. Now from the surface, these pores are what you call the gastric pits. And the gastric pits are made up of those mucus producing cells. So a lot of mucus is being produced and it is released into the lumen. As you go lower from the gastric pit, now you produce this gland. And what you find lining towards the surface here are these parietal cells. Parietal cells have an interesting shape. So we have to the basolateral side that shape, but then when they come to the mucosal side, they form these indentations so that they can increase the surface area. So we have the nucleus here. So what will happen is that when they produce HCl, it will be produced into these crypts, so that as it produces and it accumulates, then it will come to the lumen. Now, this will enable, this is actually the parietal cell is the largest of these gastric gland cells. And as it produces HCl, they are not as numerous as the others, but they produce a lot of HCl and they're increasing the surface area within which the HCl can be released into the lumen, so that the cell is producing and you're increasing the surface area within which you can release the HCl into the lumen. Now, look at the arrangement. From the parietal cell, where you produce the corrosive HCl, this HCl, remember, contents are usually produced from the blind end. And as you produce stuff here, they push contents towards the exit. So just at the neck of this gastric gland is where we have the parietal cell. Their location is very important because if you had them at the bottom, it means that they'll start producing acid, which will expose all these cells to corrosive acid without the advantage of protection from mucus. However, when they are produced at the neck, you can see that they will produce acid. And as the contents are pushed out, they come in contact with mucus producing cells. So these cells are already protecting the lining of the stomach. So you'll find they will have a very thick layer of mucus and the acid is being produced. So it ends up into the lumen without causing damage to these walls of the stomach. So as you go lower, we've already discussed the mucus producing cells, and now we have discussed the parietal cells. We are going to come across now the zymogenic cells, the ones that produce the enzyme. You can see these are the most numerous within the gastric pit or within the gastric gland. These are the pepsinogen producing cells. Now, towards the bottom, we find again large shaped cells. These are the enteroendocrine cells. Now, when chief cells produce pepsinogen, they release it into the lumen. Within these cells at the bottom, we also have some stem cells, which they have not labeled here. So the stem cells are always replacing cells and they are moving upwards. So that as you move up towards the gastric gland or towards the exit of the gastric gland, the cells will start to modify themselves and they can become enteroendocrine, or they can be modified to chief cells, or they can be modified to parietal cells. Now, when you produce acid, the acid again is released into the lumen. So all these contents are being released into the lumen, and you are having an accumulation of fluid which will be forced to exit there. So I want to show you the difference in the enteroendocrine. Remember, an endocrine gland releases contents into blood circulatory system. So this is a hormone, a hormone producing cell. So the contents are not released into the lumen, but they are released into circulating blood vessels. So if you are asked about the different cells of the stomach, you can classify from the mucus producing, HCl producing, the chief cells, and the enteroendocrine cells. And you can also add to that the stem cells that give rise to all these variation of cells. So as you cut down, as you move deeper from the lumen into the walls of the stomach, you're going to come across the muscularis mucosa, just like any other part of the GIT. Then you will have the submucosa. But like I told you, the first layer you're going to encounter is the oblique layer, which is the thinnest. But then surrounding the oblique, you have the circular muscles, and then you have the longitudinal muscles. And covering all that is the serosa. Any questions so far? Yes, Samuel.
I want to know whether the gastric pits are the same as the surface mucus cells.
Gastric pit is like a hole. It's a pit. The name pit is another name for hole. So these are the holes that are visible from the surface. They are the ones that are called gastric pit. But the walls of the pit are made up of mucus producing cells, these ones. So the cells, the epithelial cells forming the gastric pit are mucus producing cells. So does that make it clear?
Yes, it does.
Okay, thank you. Any other question? So I put this diagram here because I always forget one thing that is very important, and this is the intrinsic factor. Intrinsic factor is a protein that is also produced by the parietal cells of the stomach. This factor is very important because it is a protein. Once it gets into the lumen and you have eaten food containing vitamin B12, the intrinsic factor will interact with the B12 and it will form a complex. And this complex is the one that can be absorbed across the GIT wall. So if you do not have intrinsic factor, then you are not, although you are going to have a diet that is rich in vitamin B12, you find that you will have a problem in absorption of vitamin B12. For that reason, you'll end up having a deficiency disease. Now, vitamin B12 is normally used in the synthesis of red blood cells. So when you're having the red blood cells, it is a factor that is required for generating mature red blood cells. So when you do not have vitamin B12, you end up becoming anemic. Now, some people had suggested, or they had even tried, the fact that you are so obese is because you are eating a lot of food. So why can't we get rid of the stomach so that the food will come and without complete digestion, it can move into the GIT and will only leave a very small portion of the stomach to act like a receptacle. Now, when they did that, they realized that people who did that operation would end up becoming anemic. And that's when they started realizing there is a connection between the stomach wall and anemia, and it was tracked down to the intrinsic factor. So this next diagram here is representing what happens. We have this pink stuff here representing the vitamin B12. Vitamin B12 comes and sits within the stomach contents. However, the walls, you see this is a parietal cell, And this parietal cell, as it is producing HCl, it also produces the intrinsic factor. This intrinsic factor comes and interacts with the vitamin B12 and forms a complex. Now this complex will go down the GIT. So you remember the phases through which it will pass. Who can tell us slowly which are the phases that it will go through within the small intestines? The first step, where will it go to? Anybody who is awake? Contents from the stomach. Not Samuel, you talked, not Yesh Patel, he has talked. Yes, Anastasia.
Do you mean like the way the food will flow from the stomach?
Yeah,
so it will go to
the flow of the
duodenum?
food. Where will it go to first?
Duodenum.
Okay, duodenum, then followed by Anei. Anei, from the duodenum, where will I go?
Jejunum.
Jejunum, Asna, let me give Michelle Mogusu. Asna, you've talked. So from the jejunum, you go to? Pardon?
It's duodenum, jejunum, and ileum.
Duodenum,
It's in order:
jejunum,
duodenum, jejunum, and ileum.
ileum. Yeah, now I wanted you
And
to see
ileum.
the order. Thank you, Michelle. So it will come from the duodenum, go to the jejunum. It is still a complex. The B12 complex will go to the duodenum, go through to the jejunum, and as it goes towards the tail portion of the ileum, it is going to find epithelial cells that have receptors for the intrinsic factor. Remember, they do not recognize vitamin B12. Those receptors recognize the intrinsic factor. So by attaching to the intrinsic factor, they will allow this complex to be absorbed into the epithelial cells. Once they go into the epithelial cells, the complex will break, releasing the vitamin B12, which can then go into circulation by being passed through the basolateral site into the blood capillary. Within the blood capillary, it will bind a transport protein, and from there it will be used, or usually from the blood capillaries you go to the liver, and from there you go to synthesize the blood cells. So you see, if you do not have that complex, these cells cannot recognize vitamin B12, and then therefore the vitamin B12 will simply move into the cecum, and from the cecum it will go to the colon. So it is very important that you remember the function of the intrinsic factor and remember where it is produced from. So from there, I will quickly point, because we have a shortage of time. If I skip some of these things, we'll run short of time towards the end of the semester. So let me just start on the small intestine, although I won't have time to finish. Now, the small intestine is the longest portion of the GIT. It is made up of about 5 to 6 meters of tube, starting from the duodenum. This is the organ that is mainly involved in the process of digestion and absorption. So both digestion and absorption will mainly take place in this part of the GIT. For that reason, it is lined with cells that are responsible for secretions. These secretory cells will produce a lot of fluids, they'll produce electrolytes, they'll produce enzymes, and they will also produce mucus. For it to allow the process of absorption, we find that if it is simply a straight tube of 6 meters, the area will be very small. However, remember those circular folds that we talked about? These are permanent flow, which increase as you move further away from the duodenum. So with the duodenum, they may form small indents or small holes like that. But as you go towards the ileum, they become more peak-like structures. And as you come to the ileum, it's highly folded. This is increasing the surface area. Now, if I were to take one of these circular folds and enlarge them, what I am going to see, if I cut across like that, I'm going to see the layer of epithelial cells, and I'm going to see the mucosa and submucosa here, because the muscular lining is at the bottom. So if I take the epithelial cells, you find that the epithelial cells on the luminal side will have what we call microvilli. The epithelial cells with the microvilli are also increasing surface area here. Now, in addition, the circular folds are going to form what we call the villi. So we have this will be the epithelial cells, and they form the villi, which has a unique structure, but the villi are located within the circular folds. So I think I am not making a very clear diagram here. So what I will do, I'll go straight to where I have a diagram, and I'll show you how you increase the surface area within the small intestine. So I've mentioned the secretory activity of the small intestines, where they produce a lot of mucus, electrolytes, water or fluids, enzymes, and the other thing I had not mentioned are hormones. So I've talked about the folds and the length of the GIT, and the purpose is to increase the surface area, which is required for the purpose of digestion and absorption. So we know that the small intestine is divided into three portions: the duodenum, jejunum, and ileum, as you have already mentioned. In this diagram, these parts are color-coded, with the duodenum being purple and the jejunum being about bluish. I don't know whether that is blue or whatever color it is, and the jejunum being pink. Now, generally, this will be about 25 centimeters. It's very short, less than a meter, less than a foot, because a foot is 30 centimeters. The next portion is roughly about 2 meters. 200 centimeters is 2 meters. The next portion is the one that carries the longest length, which is about 3 meters. So in combination, you find it is just about 6 meters within the small intestine. So if you look at a cross section of this portion, we start with the duodenum. Cut a cross section of the duodenum and expose the lumen. So this is a section, the diagram towards the left is a section through the duodenum, showing you the lumen here. You have the lumen where my arrow is. Now you can see from the lumen, by the way, this whole part together with the muscles and all that, this could be representing one circular fold. But towards the mucosal area, you have those projections. And these projections are finger-like in structure, and they're the ones we call the villi. Now, you find that in the crypts between villi, you're going to have glands. Epithelial tissue will extend into the submucosa. So this will be the submucosa. You have the muscularis mucosa here, and you have the mucosa just beneath the epithelial tissue. So these are the epithelial cells, and just beneath the epithelial cell, you have the mucosa. So as you move beyond the muscularis mucosa, you go into the submucosa. And you can see from this diagram that the submucosa has some epithelial lining, and this is one area of a big gland. But this gland is not straight. It is highly branched and folded. So any of these is part of the gland, these ones. These are just highly folded glands, and the lumen all coalesce together to form a tube-like structure that will exit in this crypt here. This whole structure as it is what we call the submucosal gland, and another name for it within the duodenum, they are known as the Brunner's glands. Their work is to produce a lot of bicarbonates. So remember the location of the duodenum. It is immediately after the stomach. And therefore, all the stomach contents are acidic. They need to come and encounter a basic environment. So one of the functions of the duodenum is to have those Brunner's glands, which are going to release a lot of bicarbonates. This area of the duodenum is also the exit point for the pancreatic duct and the hepatopancreatic duct, and they exit through a valve-like structure which we call the hepatopancreatic sphincter. I don't know whether I have a good diagram that will show that, but if this is the wall and you find that the duct that is bringing in bile is the bile duct, and the one that is bringing in pancreatic enzyme is the pancreatic duct. The two come together to form the hepatopancreatic duct, and what happens, it forms a bulge like which looks like a volcano from the surface, and in the middle of the volcano is where you have the lumen that will exit the contents. This part, the bulge here, is made up of circular muscles and is the ones that will form the hepatopancreatic sphincter. So when you stimulate the gallbladder and you stimulate the pancreas, they are going to contract and eject these fluids or their contents through this hepatopancreatic valve, and they'll release the contents into the duodenum lumen. So I think that is a major structure I can talk about. Then I'll come back to the duodenum as I compare the structure of the duodenum, jejunum, and ileum. So the next structure is the jejunum. Now, the jejunum is more or less, the reason it is being called empty is that it usually does not have structures that are common within the duodenum. It doesn't have, you find that as you move down from the duodenum, as you come to the jejunum, the number of Brunner's glands start to disappear. They are fewer and fewer and fewer. So within the jejunum, we have minimal number or absent Brunner's glands. That is a characteristic, the Brunner's glands are characteristic of the duodenum. Now, as you go to the ileum, we are going to find that within the submucosa, we also have another structure that is only common to the ileum, and those are known as the Peyer's patches. So, the Peyer's patches are found within submucosa of the ileum. The Brunner's glands are found within the submucosa of the duodenum. So it is only the jejunum whose submucosa appears empty. It's like it doesn't have anything because it loses the Brunner's glands and it has not yet started to give rise to the pairs patches. So that place is sort of empty. So the other thing is that when you start looking at the villi, they start to increase in number as you go down because we increase the function of absorption. So the jejunum is the area where you find a lot of digestion taking place because the contents from the duodenum, having been supplied with enzymes and bile, as they come down through the jejunum, you find a lot of digestive processes taking place. So this is the point where we have a lot of motility, which will allow the movement of the fluids and the mixing with the enzyme and the mixing with the bicarbonate so that they facilitate the process of digestion. The last portion is the ileum. Now the ileum is the longest portion. Like I said, it is about 3 meters in length. And you find that if you are to look at the cross section from the duodenum, the lumen becomes smaller. So you have the duodenum has a very wide lumen, but as you go to the jejunum, you find that it is still large, but when you go to the ileum, it has the smallest diameter. So one way of distinguishing the 3 parts is number 1, you find that the duodenum forms a C shape around where the stomach is. So the stomach will be located here, and embedded in between here is the pancreas. So we have the stomach and the pancreas. And by the way, we also talk, whenever you think of the stomach, it's a J shape, so it has a small, I would say, curvature on one side, which we call the lesser curvature, and we have the large curvature on the other side. So whenever you draw the stomach, always remember to draw those 2 curvatures. So we have the duodenum is in the contact with the pancreas and the stomach. Beyond that, now you go into the jejunum, which is devoid of these structures, but now we start seeing mesenteries. If you look at the mesenteries of the jejunum, they differ from the mesenteries within the ileum. So how do they differ? Number 1, we have said the lumen of the jejunum is very large, and it is the one you can separate it from the duodenum because it is devoid of the tissues here. Now, when it comes to the jejunum, it has mesenteries and it has blood vessels, and these blood vessels are very elongated, forming what we call arcades and the vasa recta. So the small blood vessels are the ones which are straight-like, they are the ones which form the vasa recta, and the ones that are larger and they form a web-like structure are the arcades. So you can tell the difference by looking at the size of the vasa recta and the arcades. So for the jejunum, the vasa recta are longer as compared to these, which are shorter. Then the arcades are more modified in the ileum as compared to the jejunum. Then when you look at the lumen for the ileum is smaller as compared to the jejunum. And finally, if you look at this fat tissue, we have a lot of fat tissue. Remember that diagram I had showed you before? So if you look at the fat content within the mesenteries, it is most within the ileum. The jejunum does not have fat in the mesenteries. So this is how you can actually tell the difference between all those structures. Now, this is the gross difference. If you cut a cross section and you look at the circular folds, you find that within the jejunum, we have more of these circular folds which are thicker, but within the ileum, they start to become less folded. They are more flattened like structures. So it's like those circular folds are disappearing. And the funny thing is that they are also thinner. So you find a lot of villi within these areas, but the circular folds will be less. Now, the terminal portion of the ileum ends in the ileocecal valve. Ileo means it's coming from the ileum. Cecal is of the cecum origin. So at the junction or the last portion of the ileum, it connects to the cecum, and the valve that is located there is the ileocecal valve. So I think now, much as I want to continue, I will have to leave it at that so that I can take attendance. So kindly unmute yourself so that you can respond. Okay. Okay, so it looks like just 1 minute. So is Alice Alango here? Alice
Present,
Alango.
present, present.
Okay. Okay. Anaï?
Present.